As the screen notes, this is the fifth recorded lecture. The fifth, sixth, and seventh recorded lectures are for the week of January 24th through January 28th of 2022. Here we're continuing our discussion of the digestive system and taking a look at the anatomy and some basic physiology of the digestive organs. Actually, taking a look at some of the glands. So we'll be talking in this lecture about the liver, the gallbladder, and the pancreas. We're going to answer some interesting questions in addition to taking a look at the anatomy and basic phys, but some fun questions that I hope you can look forward to learning about, things like this. So these are questions that I might have posed earlier on in the semester or in the prior class that we had. And that is questions like, when you take a drug, let's say aspirin or Tylenol, or you're doing some kind of a recreational drug like alcohol, why do the effects wear off? What happens? So if you got a headache and you're taking aspirin, why is it that you sort of need to take more aspirin after about 4 hours? Or what causes then the effects of alcohol to wear off? And that's important too, because if you've had a drink or two and you want to be very safe as you drive and not hurt others, which is pretty darn important, you'll want to be aware of how quickly the alcohol can wear off. Some questions are fun, so maybe you've ever wondered, why is it that, politely here, feces are brown? And you'll learn that today. How about that? And finally, I'll end when we are discussing the pancreas. I'm going to distinguish between the two types of diabetes. And then it'll turn out at the end of the semester, when I lecture about the endocrine system, we'll talk more about diabetes and the causes specifically, and we'll look more about this. But I want to introduce it right now because it's such an important disease. So let's take a look. The liver, the gallbladder, and the pancreas. Here we see the stomach, and you really have to peek right behind here to see where the pancreas is located. So the last bit of what we're going to talk about, the pancreas, is located on the caudal margin of the greater curvature of the stomach. Likewise, the gallbladder, and then the great big massive liver here that's a bit larger on the right side of the body than the left, where the stomach is taking up some of that same space. So again, I'm delighted that this week, the week of January 24th through 28th, is when you are starting your look at the digestive system of a cat. There are many, many parallels, and so I'm just delighted by this overlap. Okay? Well, if we take a look first at what's going on here, this is not the lecture for the small intestine. That'll be the next lecture. But this has to do with the small intestine because the liver is generating some products, bile, that are then released from the gallbladder or directly draining into the small intestine. And the pancreas is releasing, we'll see, digestive enzymes and other fluids that are all entering into the first section of the small intestine. So although we're not talking about the small intestine today, we're talking about organs, glands that are affecting the small intestine and the digestive process that's going on. Okay? So chyme from the stomach, which is quite acidic, is coming into the small intestine, of course, metered through the pyloric sphincter into the small intestine, and then immediately secretions from the pancreas help to neutralize those acids and bring it up to a basic pH. Also secretions then from the liver, and that would be bile, that are making their way into the small intestine. It's a really important part of the digestive process, and what we've said before is that the small intestine is really where the majority of chemical digestion occurs and the majority of nutrient absorption is occurring. So let's take a look at these three principal components, the glands that are so intimately associated with the small intestine. I'm going to start with the liver, then the gallbladder, and finally the pancreas. Well, just for starters, for even people that aren't taking human anat and phys, perhaps you've enjoyed a meal of liver. Here, looks like it's being served with maybe potatoes and onions, and some people enjoy eating liver. There are organs like this that people eat, parts of the digestive tract, the stomach we talked about before. And in general, when people are eating meat or some kind of meaty product from probably a cow or maybe a pig, generally those are muscle. But in this particular case, it's a digestive gland. And that's what it might look like raw. And I happen to know a lot about this raw beef liver because for a very long period of time, we fed strips of beef liver to axolotls. And it turned out to be a really good source of nutrition. And in fact, it caused problems because sometimes it was too much of a good thing. And sometimes these animals, our axolotls, got too much vitamin A and perhaps too much iron just as well. So eating pretty exclusively beef liver could be a problem, but it has been something that people have done with axolotls. I've chosen instead to go with earthworms as a more balanced and natural diet for them. Okay, where do we find the liver? Well, we find it just below the margin of the diaphragm. And where's the diaphragm located? Well, it's dome-shaped and it's cupped here along the margin, the caudal margin of the ribs. So the diaphragm is located in here and the liver is pressed right up against it. Over here, occupying some of this space, is the stomach. So it's not equal, it's not symmetrical. The liver is reddish brown in color, and I'm just curious if you have an idea of why it might be reddish brown in color. I'll give you a hint. The color of the liver is very much the color of the spleen, which we have not talked about yet because the spleen is not part of the digestive tract. And in both cases, the liver and the spleen are this dark brown because of the heavy investment of blood. These are blood-soaked organs, and we're going to talk about that in just a couple minutes about the blood distribution within the liver. So by far the liver is the largest gland of the body, and it does so many things. Now we're only going to talk about the digestive gland functions, but you should know it is a very long list if you just considered the liver proper and all that it does. So here, regarding the digestive system, we're going to talk mostly about its secretion of bile, but it's also storing carbohydrates that are extracted from the blood that's coming straight from the digestive tract, and it's also breaking down toxins. So we'll talk a little bit more about some of these other functions, but we're going to focus on those functions that are related to the digestive tract. Okay, so those functions not directly related to digestion will be addressed in other chapters. So now a little bit of the gross anatomy. You're able to study this in a cat, and you should get started on that this week, so that's lovely. And these are generally the same lobes, although it might be divided up a little bit differently in a cat. But there are right and left, and then quadrate and caudate. So don't get confused by these. Quadrate and caudate lobes. Now, for starters, the falciform ligament is running right down the middle, dividing right and left, but again, those are unequal in size, with the right lobe clearly much larger than the left. And so what is the falciform ligament? It's this mesentery that is dividing the liver. At the very caudal edge of the falciform ligament is something that's called the round ligament, which is a remnant, so it's a bit of a scar tissue of where the umbilical vein was running when you were a fetus. So the umbilical vein was carrying then blood from the umbilical cord to the liver. And here we see those structures, the round ligament and the falciform ligament in this artwork. Here's the falciform dividing the liver unequally into right and left lobes. By the way, this is gallbladder over here. And then the edge here of the falciform is that round ligament, which was carrying blood to the liver from the umbilical cord. Here now, cadaver highlighted, and I hope that you like this artwork, this particular photograph here of cadavers, because it's a nice example of the reality. Here you're not relying on an artist interpretation, but you can see the real thing. So I think that's really good. Here you can pause this and study this in detail. So that's again why this is a big advantage for these recorded lectures. I understand that this is something we're doing because of the COVID situation, but I really think that this is an advantage. So you could pause this and look at these in some detail. And it's nicely identified and it's real, and I think this is going to be more helpful for those of you that go on and have a chance to study human cadavers. Here the positioning of the liver. You can see in the upper left. Here showing you the gallbladder that's nestled within the liver. And I think it's important to note at this point that the liver very much conforms, that is, it gives in to the shape of the structures around it. So the liver is kind of like something that would be a really well packaged product, maybe coming from Amazon, and that doesn't happen as often as I'd like. But here, if they pack things very tightly, the liver has taken on the shape of the surrounding organs. And as you'll see in the cat, the very caudal margin of some of the lobes of the liver actually take on the shape of the kidney, and so they're impacted by the shape of the kidneys, as well as the gallbladder and the stomach. Of course, that's going to be, well, this is the stomach right in here. The microscopic anatomy of the liver is complicated. The liver is a very, very complicated structure, and you may have studied already a few sections of liver as you were learning about epithelia, because this is a simple cuboidal epithelium. But the arrangement here is altogether different. Now, the textbook uses the term filtering, saying that the liver filters. But I want you to know that when I think of a filter, I think of something like a coffee filter. Okay, so there's going to be some paper and coffee runs through with coffee grounds, and it's going to catch the coffee, and it's going to allow the grounds to stay back and allow the coffee to flow through. And here's the important part. When you're done making coffee, you have to get rid of the filter, and you're going to start over with a fresh, new filter the next time you make some coffee. Well, that's not what the liver's doing. We don't need to have a liver change. By the way, that coffee filter kind of mechanism is what's going on in your car with an oil filter, okay? And we change oil filters. So I don't want you to confuse the terminology used in your textbook of filtering, to think about it filters and captures particles. Instead, it percolates. That's the word I'd rather have. It percolates. It flows through and past cells. That is, blood flows through and past cells. So here we've got a branch of the hepatic portal vein, which is taking blood. This is draining blood from the gut now directly to the liver, and here we're seeing those veins branch out. There's also a direct branch of a hepatic artery that contributes, and it contributes nicely oxygenated blood that keeps these cells alive. And so those two sources of blood are mixing together. Blood with lots of nutrients that have been absorbed from the gut, and blood that carries lots of good oxygen that keeps the liver cells alive. And it's this flow through system. So blood is flowing through and into this central location. And so the textbook, your textbook, uses an analogy about a book. And they said that if you put a book on end and spread out all the pages and sort of took the cover of the book and matched the cover to the cover so that the whole book's pages fanned out into a circle, they said that would be a bit like this arrangement. Okay. Well, I don't think this is too difficult to understand. What we have are channels that are all converging, right? So you can see them all converging. And so there turns out to be a functional unit of the liver right here that's called a hepatic lobule. A hepatic lobule. And a hepatic lobule is then the functional unit right here of the liver. Now, we saw something like this when we looked at bone. Okay, now, we're going to talk about this in lab as well. But we saw something like this when we looked at bone. Bone consisted of these kinds of functional units. And do you remember what they were in bone? And that'd be great if you did. It was an osteon. An osteon. So if you remember what was going on in compact bone, we have these functional units like this. Blood vessel was in the middle just as well, and we had rings of bone cells. Here again we have a blood vessel in the middle, and in this case the organizational unit, a hepatic lobule, is different, but they're all sandwiched together. So hepatic lobules are next to other hepatic lobules, just like osteons and compact bone, and collectively that forms then the liver. These hepatic lobules all scrunch together. So you can see some connective tissue that separates out the hepatic lobules. So what's happening here is that the blood is flowing peripherally to centrally, so from the periphery to the center. And as it does, that blood is flowing past these hepatocytes. And these are the main functional cells of the liver, hepatocytes. Now the next slide is going to show us some more detail of the same figure. This is 2520. It'd be great to have this out. What you can see then are these hepatic sinusoids, the blue-colored tubes here, are then the venous blood, but with a little arterial blood thrown in, and it's flowing past these hepatocytes. Now, over here in this up close on the right side, they're giving you a detailed view of exactly what does that flow-through system look like. And for starters, the blood vessel is, as we would expect all blood vessels to be, it's lined by a simple squamous epithelium that forms an endothelium. Okay? So this is the endothelial cells, and that's actually what that label says. You might have to enlarge this image or look at it in your book. So here are the blood cells that are lining the blood vessel. The cells that are lining the blood vessel. Here are blood cells that are flowing through. And then take a look at the nature of the hepatocytes when they're underneath some of these endothelial cells and also in between endothelial cells. And there's gaps in the cells here. These hepatocytes have microvilli. Now, why do you think these hepatocytes have microvilli? What's the functional significance of microvilli on hepatocytes? And I hope you can guess that it's to increase the surface area in contact with the fluids, the plasma, that's flowing past these hepatocytes. And so these hepatocytes can chemically interact and pull things in then, have things selectively brought in or diffusing in, and they get processed within the hepatocytes. So we'll talk about functions of hepatocytes here in a second. But that's an intimate relationship between the hepatocytes and, again, the plasma, where most of the substances that are carried in blood occur within the plasma. Now, this is what you're going to see in lab, and this is what you're going to need to interpret in lab. And boy, oh boy, there's a lot of histology for us to study as we get going here again and take a look. So this is one hepatic lobule. Here's the central vein. Okay, here they identified hepatic lobule over here. Here's a branch of a hepatic vein. So there's a bile duct, and it shows you a branch of the artery. And maybe I've said this before, maybe you learned this before, but the arteries are going to have stiff walls and be quite thick. veins have thin walls and they often do not have a nice circular shape. And then over here, the bile duct has a different kind of lining altogether. So it has a different type of epithelium inside. And you'll need to be able to identify all three of these structures when you're looking at a tissue. So when we're studying the histology of the liver. When you look at a liver lobule like this, one of the things that catches my eye is that you do see these lines, these radiating lines that are converging in the center of the lobule. You don't see them everywhere, but where the tissue is cut properly, you can see that. And I've said to you before, maybe in Human Anatomy Phase 1, in this last fall or whenever you had it, that one of the things you always want to do, I mentioned this in zoology as well, when there's an expert, and I'm an expert on histology, when there's an expert and the expert is telling you how they identify things, pay close attention, because sometimes there's little tricks. But this is one of the triggers for me. If I see these series of converging lines right away, boy, oh boy, there's very few other tissues, I can't think of one right now, that have this arrangement other than a hepatic lobule. So if you don't know what tissue's in front of you, boy, this is a good sign. Now here's higher mag that they provide, and you notice again these converging hepatic sinusoids, which is where the blood's flowing through, and then these are the hepatocytes. These are the liver cells right here. So what they've done on the right side is to highlight these things. And what's kind of interesting is these cords of hepatocytes, and then right here, the sinusoids, all kinds of has the same pattern. And sure, that's because these hepatic cells, hepatocytes, are arranged in sheets, and then there's streams of blood between them. So just like fanning your fingers out right now, your fingers could represent these cords of hepatocytes, and then the space between your fingers right now could be like hepatic sinusoids where the blood is flowing. And if your hand is still fanned out, imagine that the space between your fingers is all converging in the center of your palm, because that is what's happening right here, where the blood then collects in the center of the hepatic sinusoid. So this will be good for you to maybe review even this lecture when we do some of the histology. We'll go into more detail when we do this in lab. Now, on page 965 of your current text, so I've confirmed that this is the 8th edition, there's a long list of what the liver does. So I just want to run through some of this right now and make a couple comments. Remember that hepatocytes are most active after a meal. Think about why that is. Well, after a meal, a few hours after you've consumed a meal, some of those nutrients then are being selectively absorbed by your digestive tract, mostly in the small intestine, and that blood immediately heads to the liver in a very low pressure system called the hepatic portal system. And so right after a meal, that is a couple hours after a meal, these hepatocytes are suddenly flooded with all sorts of nutrients or whatever it is you've taken. If there's toxins that you've taken in, these hepatocytes are going to use chemical reactions to chemically neutralize toxins. So there's going to be absorbing of glucose, all these other nutrients, vitamins, minerals. And then what happens is either some of those nutrients are broken down right away, and we generate then hopefully ATP. That's a common form of a functional molecule. As we said before, it's a currency. Or we store, and there's an awful lot of storage of going on of these nutrients. So I make the analogy to going grocery shopping. When you come home from grocery shopping and you haul in your groceries, you make a decision. Are you going to eat it right now, or are you going to store it? And you would probably have to go to the store pretty often if you bring things home and eat them right away. So most of the time we go to the store and maybe eat a little bit, right? But we store for the next few days. Hepatocytes remove and degrade all sorts of toxins or things that need to be broken down. So hormones, toxins, could be chemicals that we've taken as a medicine, bile as well that's been released by the liver. Eventually in the bloodstream comes back, and the hepatocytes are going to break it down and recycle. There's going to be some pigments, and then I've mentioned already drugs. So the answer to the question of why do you need to take aspirin again after 4 hours is because that aspirin gets absorbed into your circulatory system. It circulates throughout your body. So although you might be taking aspirin because you hurt your knee, that aspirin is affecting all the parts of your body, all the different areas of the body. It just happens to provide additional relief to pain in your knee if that's the case. But that your hepatocytes are always taking a whack at that aspirin. And so once it's absorbed, the hepatocytes are starting right away to chemically break down. Do not think of it as filtering. Chemically, biochemically breaking down the aspirin. And that's why, likewise with alcohol, alcohol wears off. And when you think about it, and it varies, by the way, alcohol wearing off has to do with how effective your body is at breaking down the alcohol. And that would have to do in part on your body size. So your body size is going to have then proportionally a larger liver if you're bigger. And if you have a larger liver, you've got more hepatocytes to break it down. So let's say it's one drink. And so I believe it's about a regular beer is about equal to, 4 ounces or so of wine. You can check the alcohol contents. And that's equal to about 1 shot of some kind of hard liquor. In general, a healthy person of pretty much average size can probably break down about that amount of alcohol every hour. Okay, so if you had one drink and it's quickly absorbed, your liver can break it down over about an hour. There are charts and things that you can look at, and I don't want you to trust your life on this, and I don't want you to risk other people's lives by going out and driving and assuming all that's true. Some of you might be smaller than others, and further, how good and healthy is your liver? So if your liver is doing this a lot with drugs, it might become less effective as the liver itself gets damaged by the alcohol or other drugs that you're consuming. So it depends on your body size, depends on your metabolic rate, depends on the health of your liver. But nonetheless, these hepatocytes are chemically breaking down all sorts of drugs, including alcohol. So lots of things that hepatocytes do. And again, a lot of this is listed in the chapter. So making albumin. Albumin is that, as you will learn about other times, it's important in many different ways within the body, but albumin is the white part of an egg. Lipoproteins, some clotting factors, factors affecting blood pressure, angiotensin, and breaking down glycogen and storing it in the form of glucose and entering circulation. So we'll store sugars when we store them as glycogen in the liver, and then in between meals, you can keep your blood sugar level up by breaking down glycogen. So here's another question I could have asked at the very beginning. In between meals, you might be anticipating another meal. You might be anticipating a meal right now and sort of feeling hungry as we talk about all this. And I don't know if you've concentrated on this, but if you're really hungry and you push through it and you don't eat, your blood sugar level is going to be dropping. But eventually, you might notice that you sort of get past the meal. And that's because when you get really hungry, it stimulates the breakdown of glycogen that brings then back up your glucose levels. So your liver can respond to your hunger pains and actually help to provide a higher glucose level of your blood and decrease your hunger urge. Okay? So that's how we keep steady blood sugar levels in between meals. There are also macrophages within the liver, and these are removing bacteria, and the book says debris, but you should know debris could be any parts of cells that have broken down. So removing damaged cells, generally cleaning up. Sometimes we think about that as a function of macrophages. And of course, they produce bile, which is what we said is this really important fluid that helps to break down fats in our digestive tract. And that bile drains first into these little ductules. They were highlighted in some of the other figures. And then finally into the common bile duct, the common hepatic duct. And then into the common bile duct. So I'm going to talk about bile and the gallbladder here next. Okay. Here's a good image, figure 2521, showing you then the basics of the relationship of the gallbladder to some of these ducts. So there's the hepatic ducts, there's the common hepatic duct, and then this is the common bile duct right here that then is coming down and providing those fluids, metering them into the small intestine, and it's actually the duodenum, which is the first section of our small intestine. So it's this pear-shaped kind of structure, stores and concentrates bile. And what is bile? It's a kind of bitter tasting, this yellow-green kind of fluid. And look at all the things that are in bile. So minerals, cholesterol, fats, phospholipids, pigments, and acids. And these are really important in helping break down some of what it is we've consumed. Okay, so here's that figure in close-up. You can see how the bile duct comes down and joins one of the ducts here from the pancreas. And so this particular area is a place that's regulating secretions from the bile duct, as well as one of the main ducts that's coming from the pancreas. Now, I'm going to mention this later, but so I'll probably say it again. Over here, notice that there's a side branch off of the pancreatic duct, and that duct is not regulated like this, and it allows a regular, steady flow of pancreatic secretions into the duodenum. So we can have a regular flow of pancreatic secretions, and then after a big meal, we can have a big dump of pancreatic secretions and bile. Okay. Why did we talk about the gallbladder and the bile ducts and everything in zoology? Does anybody remember that? And the answer was we talked about liver flukes. And I said that the liver flukes, which are acquired through food, make their way up here through this opening and live in the bile ducts. And that's a pretty foul fluid to live in, but nonetheless, that's where these liver flukes are living. And then they can make their way up all the way through the hepatic ducts, and they can even start clogging up hepatic sinusoids, and that can lead to then liver failure and could be life-threatening for some people. Yeah. So bile contains lots of things. Contains bilirubin, which is a pigment derived from breaking down hemoglobin. So when we said bile contains a pigment, here's one of the pigments called bilirubin, and it's pretty important. And it helps to break down hemoglobin. Then bacteria in the large intestine are metabolizing bilirubin and converting it to urobilinogen right here. And that's what makes feces brown. So ultimately, why are feces brown? And it's a byproduct of the breakdown of hemoglobin. Yeah. These bile salts, these acids, are steroids that are made from cholesterol. And in your book, they talk about the regular cycle here of making bile salts, and then their recycling of some of these bile salts in the intestines, so they're reabsorbed but not all are reabsorbed. And so there is some loss of these bile acids, these bile salts, and that is a way that we get rid of cholesterol out of our body. And it has to be replaced. So what are bile acids and lecithin doing? Lecithin, what is it? It's a phospholipid, and they're helping to break down fats and absorb them. So the secretions and bile waste are eliminated, but I want to go back to this point. So what's going to happen to somebody who's had their gallbladder removed? So you got to think that through. Does it mean that they don't have any bile that's going into their small intestine? And the answer is no. They will have bile being released into their small intestine, but there won't be an accumulation, a large accumulation of bile in the gallbladder. And that means if the person consumes a very fatty meal, they're going to have trouble breaking down the fats and absorbing them. And so we would predict then that if you lost your gallbladder, if they had to remove it, let's say, which is very common, then if you eat a really fatty meal, whatever that might be, but it's probably a lot of fried things, then we would expect a lot of those fats do not get absorbed, and instead the fats continue through your digestive tract, and they are going to contribute then to a fatty kind of diarrhea. Okay? And that probably will cause intestinal cramping and not be very pleasant. So, it's my general understanding that if you've lost your gallbladder, you can still eat fats, but you eat them in smaller proportions, and you will keep track of how you react to all that and be able to moderate that yourself. Okay, so then other secretions in bile are things that are being eliminated. And finally, gallstones, which can form in the gallbladder, of course, are accumulated waste, things that don't get released out but somehow are chemically reacting within the gallbladder and forming these stones. So how do we get bile into the gallbladder? And it shows right here. First, you're filling the ducts, and then the overflow goes into the gallbladder. And this is a lot, guys. A half to 1,000, or 500, well, half to 1 liter, so that's 500 to 1,000 mLs of bile made daily. And that's a lot. That's a liter. So it's half of a 2-liter bottle, right? That's pretty big. I don't know. Maybe you're drinking out of 1-liter bottled water and you have an idea of what 1 liter looks like. But that's about a quart. That's a lot of bile. Here, we're noticing that about 80% gets reabsorbed and recycled, but 20% are excreted in the feces, and that is what I said was a way of getting rid of some cholesterol and lowering cholesterol. I think I saw a note in the book that there are some drugs that help to moderate cholesterol levels of a person if they're too high, and they interfere with the resorption of cholesterol here as we're preparing feces. So if you interfere, if you reduce this number, let's say to 70%, then you would be eliminating a higher percentage of cholesterol. And that would have the effect of lowering the cholesterol levels in your bloodstream. So the liver is extracting cholesterol, and it's helping to make them bile. So here's some of those cadaver photos with the highlights. Highlighting here first the liver, the gallbladder, and then if you zoom in on some of these, so I'll get the cursor out of the way, you can see here's where then the pancreatic duct and the bile duct together are emerging within the duodenum, the small intestine. So this is starting to put things together a little bit. Doesn't have the liver. The liver is assumed right up here at the top of this diagram. But now we're going to talk about the pancreas. So it's kind of a spongy gland. It's retroperitoneal, so it's behind the peritoneum. Its kidneys are just as well. And I said before, it's caudal to the greater curvature of the stomach. So find out where your stomach is, which favors the left side of the body, and this thing tapers. you can see that the tail is over near the left side of the person's body, with the head way over here, the expanded part, the head indicated on the right side of the body. The interesting thing about the pancreas, and we've looked at it probably in cell biology, is that, and in human anat and phys, it's a gland that is both endocrine and exocrine. So that means that one part has ducts, D-U-C-T-S, and the other part releases its product into the bloodstream. And I hope you remember which is which. Which one of these has ducts? And I hope you remember it's exocrine. Endocrine releases into the bloodstream. The endocrine portion of the pancreas is helping to regulate blood sugar levels. They're releasing hormones that are helping to regulate blood sugar levels, and we'll talk about that when we get to the endocrine system at the end of the semester. The exocrine part is much more involved with digestion. Okay. So here's the endocrine part that I just talked about. How about the exocrine? So most of the pancreas, by far, 99%, makes what's called pancreatic juice. And that's kind of a general term. It's funny, in all the technical things here in human anat and phys, we'll just call it, it's pancreatic juice. Okay. So what's happening is that these glandular parts that form the bulk of the pancreas are releasing into ducts that all are converging in, as you would expect, the pancreatic duct here. Pancreatic duct runs right through the middle of the gland, and I already talked about this accessory pancreatic duct, but here's the description about it, which is it's a bypass. The accessory pancreatic duct is a bypass of the sphincter here that controls the main pancreatic duct and the bile duct. We call it common bile duct in the cats right here. So that this big surge of bile and pancreatic juice can occur as the chyme is released from the stomach into the small intestine. But otherwise, there's a regular flow of pancreatic duct here. And that's helping to buffer the pH here, to always keep it in that basic kind of range. So pancreatic juice, table 25.2. On 970 is going to tell you that this juice has a lot of things in it. It's alkaline, as I said, so it's basic. Lots of enzymes. Zymogens, if you don't know, are inactive enzyme precursors. So zymogens are something that has to be converted into a chemically active enzyme. Here's buffers. And, of course, all kinds of electrolytes, keeping things pretty steady. And so here's their components that we'll see in the next slide on table 25.2. And we're not going to spend time on the regulation of the secretion of these kinds of products. It's going to be too much. So sodium bicarbonate is neutralizing, so we have an acidic content of the stomach. That acid is helping to kill all kinds of pathogens that might come in with our food. But by the time we come to the enzymes and all the chemical conversions that allow us to absorb nutrients, we do it not in an acidic environment. It's in the small intestine, occurring in a basic environment. Here's some of the other components of things that we talked about, even breaking down DNA and RNA. Here's the cadaver images showing you the pancreas and the liver, parts of the liver here. Here's probably part of the stomach, the very beginning, the cranial end of the stomach that's been cut here in this cadaver. And here we have a cutaway of the small intestine. So we can see this pancreatic duct that's coming in and dumping. Here the pancreatic duct is highlighted right here. The three regions of the pancreas, simply tail, body, and head, from left to right, tail, body, and head, are identified here in the bottom. Here's taking a look now, of course, at a cross section. So as you're looking, one of the orienting ways or markers for you, the orienting markers, is this T-shaped vertebra. So this is the neural arch. There's where the spinal cord would be. There's the centrum of the body of a vertebra, neural spine. So you know that this is the person's back. Okay. I'm going to guess that these are kidneys, actually cranial ends of the kidneys here. I think this is probably part of the liver, and over here probably part of the spleen, I would guess, is right over here, but they don't identify it. So, microscopic anatomy of the pancreas, and you might have seen this before. So, this is the part that we said is about, the whole collective exocrine is about 99%, and it's not all secretory units, but these are the secretory cells within the pancreas that are the, of course, exocrine part of the gland. And then here we have a bit of a duct that's making its way down into the main duct that's carrying these products that are all converging onto that main pancreatic duct. Okay, so here's a little duct and here's a bigger duct. We're going to look at some of the details of this histology when we do this in lab. But here, looking at this section, here's the general image, light microscopy, and here they identify, and they're identifying with blue. Again, they're highlighting with this kind of, I don't know, it's a kind of light blue, whatever, turquoise maybe. These are pancreatic islets. These are the endocrine portion of the pancreas. And here in all this massive blue, this is then the exocrine portion of the pancreas, which we said was about 99% of the pancreas. Okay. Now I want to talk just really quickly about these pancreatic islets right here and diabetes. Okay. Now there are two main types of diabetes. We will talk about this again when we get to the endocrine system at the end of the semester. Type 1 diabetes is something you're often born with or occurs early in your life, and it's generally something that's caused the beta cells to die. The pancreatic beta cells, and these are the ones that make insulin. And so people who have type 1 diabetes are often born or acquire very young in their life, and they require typically insulin injections. So we have to make insulin now. We used to extract it, I think, from cows' pancreases. But now we can produce it directly, and we can inject it on a regular basis. So a person has to monitor their blood sugar levels and inject. But guys, here's where we're coming. Here's where we're on the cutting edge of this right now, and you should know. Look, January 17th this year, we have been experimenting for the past few years with transplanting pancreatic beta cells into a person. Now the problem is, if we transplant cells that are not matching the immunology, the markers of your own body, then your immune system might reject them. And that was unfortunately what's been happening. Now some people are describing, and this actually was in Illinois, Northwestern University as I understand it, are trying to encapsulate these beta cell transplants in ways that the immune system will not reject them, and that these transplants would be able to measure glucose levels. So that's what these cells are doing. They're reacting to drops or increases of glucose levels, and then they can on their own release more insulin. So if we can successfully transplant beta cells into people with type 1 diabetes, and we can prevent the immune system from attacking them, we could essentially cure type 1 diabetes. That's very, very, very cool. So, who knows, maybe it's something you'd like to work on. Now, you might know there's more than two types of diabetes, but type 1 and type 2 diabetes. You hear about type 2. And type 2 diabetes is the problem that when we have glucose circulating in our blood system, our cells of our body are not taking up that glucose properly. And so the glucose level remains higher in the bloodstream. And this is generally something that happens to people over the age of 40. And it seems to be impacted by a poor diet and a lack of exercise. So this is one of the many reasons why my wife and I take regular walks. Bragging here, but our walks are about 2.5 miles long with our dogs. And we took 235 walks last year for then more than 500 miles just last year in 2021. And as soon as I get done here, we're going to get some lunch and we're going to go for another walk on here a very cold day. So we don't let that stop us. Why? Because I've said this before to you guys. You need to understand that good health and good quality of your life is largely up to you. You can take care of yourself. You can eat properly. You can exercise. You can deal with your stress. You can deal with mental health issues. And whatever health you have, you can keep it up. Okay? Each of us faces our own other health issues, right? There's all sorts of things based on our genetics, for example, or our environmental circumstances or accidents or whatever can impact your health. But there are things you can do to feel better. And it can be tough, but I'm telling you, this is what we know. Eat a healthy diet, exercise on a regular basis. It doesn't mean you have to go to the gym. Walking, a brisk walk, it can be very helpful, and I encourage you to do it. I'll just tell you a quick little aside here, and that is in 2004, it's sort of when I turned things around, so it's 18 years ago. I went to an international conference. I was invited, presenting a paper. That was pretty cool on comparative anatomy. And there I met two of my mentors who I hadn't seen in over a decade. And when I saw both of them, my heart fell, and I thought, oh my gosh, these guys are fighting cancer or something, because they both were much thinner than I had known them. And then I talked to my dear colleagues, my dear mentors, and I found out both of them had deliberately lost weight because as they got a little bit older and their systems were a little more stressed, they decided as smart people that they would do this. They would watch their diet and exercise and really watch their weight. And it was that meeting that I thought to myself, so smart, educated people like you here at college, should be smart enough to control your body weight and should be smart enough to realize that they have a responsibility to themselves. And I came home, and about 18 years ago, I started exercising, and it's been nonstop, regular aerobic exercise ever since then. I had already been doing it for 6 years prior, but I hadn't been watching my weight. And I'm now at least 40 pounds lighter than I was at that time. Well, guys, if that wasn't enough to convince you to take care of yourself, how about this? Type 2 diabetes used to be a disease of aging, and now we are seeing it in kids. We're seeing it in teenagers and 20s and 30s. It is no longer a disease because of the poor diet and the lack of exercise. And I've said this in Human Anat and Phys, one in the fall, and I'll end here because this is the last slide talking about this. I'll tell you this, guys. Look, here's reality. Most of you are just maybe in your late teens or in your early 20s. And after you graduate, you're going to start jobs, and many of you will start families. And when you do, you will have a tremendous number of responsibilities. You will be young in your career. You'll be under pressure to perform. And you'll be under pressure to take care of your family and all of those you love. Financial pressure, paying off student loans, all of that stress. Who has time, Zalisco, to exercise? Who has time to eat a proper diet? I'm just going to stop at McDonald's on the way home or eat some other fast food. I'm going to wake up in the morning and have a Twinkie or a Pop-Tart or something else because, darn it, I'm too busy. Yeah, well, that's how you put on the pounds, and that's how you start developing type 2 diabetes. And you need to know that that isn't an excuse. And what I'm saying is, you simply must make taking care of yourself as high a priority as taking care of those you love. Because if you're not healthy, you're not able to take care of those you love as well as you might like. So taking care of yourself helps take care of those you love. And I leave you with that. We'll talk more about the endocrine things later, but this is the end of the 5th recorded lecture.